standing at one of the oldest confirmed academic sites in the world. There's not much left, of course. It's the Lyceum of Aristoteles in Athens, Greece. And of course, since we're going to talk about aging, I thought it would be fitting to show this image, because this didn't age very well. But now, actually, I think we have more and more means, for humans at least, to actually improve aging and to prevent them from deteriorating very fast. So the title of my talk is How to Age Well, and I will focus on process of senescence as the center point. Aristotle also had some quotes on aging, and back in those days the ancient Greeks already had quite some ideas on how to age well. So they knew that if you modify lifestyle and you don't eat too much and exercise well, you had a good old age. But still, even then they said, hope is with the young, for the young have the future. And I will show you in a minute that this may not entirely be true anymore. At least in model systems like mice, we are able to stall the aging process quite nicely and maybe even revert it. So for today, I will present you my triad of topics. I'm very much interested in the molecular causes of cellular senescence, but aside from the question how cells become senescent, we know that senescent cells are the root cause for some aspects of aging and some aspects of cancer. So even though I've been working on this for about a decade, and the field is even older than that, we still have only scratched the surface. So for the coming years in UMC Utrecht, I will focus on identifying the molecular hallmarks of what makes damaged cells senescent. And once we know, we can then hopefully design better treatments to eliminate the deleterious consequences of these senescent cells, or even eliminate those cells altogether. So of course the question I often get when I say I study aging is why? Cancer is a disease, people die from it, right? Aging is a hobby for the rich. I will show you it's actually the opposite. A lot of the diseases we get at old age. So I don't think aging is a part of life necessarily. Yes, the circle of life is of course that we get born, we thrive in adulthood, and then we deteriorate. But why is it? And what if we can stall that? And the reason I believe this is important, maybe even more important than research in all the individual diseases we do here at the hospital, is because of the following. As you can see in this graph, if the pointer works, okay, so you can see that we keep getting older. So this has been going on since the Industrial Revolution. So since the 1850s, every generation gained about 5 to 10 years over the previous generation. At least here, for instance, as you can see, for the last decade, 2 decades or so, that we've been mapping it also for Western Europe, you can see that indeed we gained about 5 years over the last generation. This is great. We actually plot here the life expectancy for women. The reason we usually do that for women is because men get in all sorts of accidents and wars, and it's not so easy to plot anymore. But the idea is the same for men. So we keep getting older. However, we keep getting older because modern medicine keeps us alive. So we get older with pills and syringes and all that kind of stuff. So if you look at the number of years we spend in poor health, meaning 2 or more age-related diseases, this also goes up. So Italy, for instance, is a country which in the EU was always the best in terms of health span. And you can actually see that you have these blue zones in Italy where people have healthy Mediterranean diets, they have a laid-back lifestyle, they have social support, and they had only 11 or so bad years before they would die. But even that has gone up dramatically in the last several decades. The same is true for the Netherlands. So we get older, but in a bad way. And it has been calculated by the econometrists that I've been talking to for the European Union that by the year 2040, actually a bit sooner, 2035, we have a break even in the cost for healthcare that we can still sustain. Beyond that, the silver tsunami is becoming so large, we can no longer afford health costs. So we need to act. And then the question is, of course, why? Why is this? Why are we getting so old with all these pills and syringes? And that's because kicking in an open door, as we say in Dutch, we get older, but we get also all these diseases. So things like cancer, cardiovascular disease, infectious diseases, we are all susceptible to these at old age. You don't get them at the age of 20. There are exceptions, but of course, these are all age-related. So if you are able to target the root of the problem, the aging of cells in our body, we have a solution to all of these individual diseases. And that's why research on aging as a whole is more important, in my opinion, than the investigation of these individual diseases. And then, of course, the question is, if we understand all this, can we then develop ways to counteract the deleterious consequences of aging? And to address this, we need to first ask ourselves a very simple question, but a hard to answer one. Why is it that our bodies age? We can see on the outside that we get wrinkles and gray hair, etc. But what is it on the inside of our bodies that makes the cells in our body age and the molecules therein? And also this is, of course, partly known from textbooks. We know that being alive, we accumulate damage. Our cells accumulate damage to proteins, lipids, DNA, from alcohol, UV light, food, etc., etc. And at some point, DNA or proteins can break and they have to be replenished. And this usually goes very well, but not always. So this is, in a way, how I see the role of damage throughout life as a contributor to age-related pathologies. I drew here two cells, two young cells, actually it's the same one. So on the left it is healthy, right? And then there's a few types of damage that can occur either on proteins, DNA or lipid, etc. And then if that occurs, there's a lot of repair mechanisms in our body. So this is what you learn in textbooks. There's repair, and if it's too extreme, the cells die off and the stem cells differentiate and make new cells. This goes very well for the first 4 or 5 decades of our life. However, as we go older, more and more of this cell stays behind. So the cells can become scarred. They really develop scars in that regard. And at some point, throughout time, but it can also be enforced by extreme stress. For instance, if you know children that have leukemia, they are given chemotherapy. They're cured from the cancer, but the chemotherapy makes them age very fast. So things like extreme DNA damage by chemo or radiotherapy can accelerate this whole process. Something which you also see, for instance, in progeria patients that at the age of 10 look like they're 80 years of age. That's because they fail to repair their DNA. But for the most of us, this is something which happens throughout normal aging. When there is too much damage, at some point, this reaches a point of no return where this becomes irreparable. And then the following happens. All the damage clusters together inside the nucleus of a cell and you get a remodeling of the DNA. This we now call heterochromatin. And then all the other damage starts to accumulate around the nucleus. So throughout my talk, I will use this cartoon of a senescent cell to indicate when the cell has become senescent. What happens is such an irreparably damaged cell becomes dormant. It can no longer divide, which is good because these are very bad cells. They're damaged cells. You don't want them to divide. However, if you have too many of them, you can see you can look at them as being rusty. If you have too many of these cells, you basically get rust on your body. Okay, so why do we age? There are many hallmarks of aging, but a lot of them are in the realm of speculation. Some of them are more proven than others, but at least the things that are very well studied and very well proven is that there's a lifelong accumulation of damage, causing cells to become senescent, and this is known to drive physical decline. So I present you this now as a matter of fact, and of course as a scientist I'm not at all satisfied with this explanation. Because why? Why are senescent cells so bad? It's not the rust. I mean, I cannot imagine that you just have some rust on your body and then your body doesn't function very well. So there must be something else. Again, I drew two cells here. Now there are different cells. And I also drew two icons on top of them. And that's how cells communicate to each other. So they can do so through direct contact, but I'm not focusing on that right now. Healthy cells secrete things to their neighbors, and that's how they talk to each other, and that's how they know when something needs to be modulated. However, if now, let's say this bottom cell becomes senescent, as again with the blue color around the nucleus, you see the following occurring. This is not at all a dormant cell. So yes, it stops the cell cycle, it can no longer divide, but it's actually shouting for help all day long. So it produces these proteins and metabolites and lipids, which together have a very complicated name. We call them SASP, and it's the secretory phenotype associated with the senescent cells. So Senescence-Associated Secretory Phenotype. So again, a healthy cell, when it becomes irreparably damaged, it starts to secrete all these factors to the environment. And this has a negative impact on the direct neighbors. See this as, you know, in your home, you have a neighbor that is very noisy all the time and drinks a lot of alcohol in the garden. At some point it affects you. You didn't change, but your neighbor made you change, right? So this is something senescent cells are those terrible neighbors. So this cell is negatively affected by the senescent cell. There's more though. again, if we now take the last image of the previous slide and we place it in a certain organ, let's say fat. And this is an experiment people really did in mice. So they took young cells from mice, they made them senescent by what I told you what you can do with chemotherapy or radiotherapy, so extreme amounts of damage. You can do that in a lab. So you take healthy cells, make them senescent, and you put them back in the fat. You get this situation, indeed, in the fat you have all these disturbances. But what else is there? They found that these factors start wandering around through the body. And indeed, what they observed is that also distant organs are affected. So if you have a lot of bad cells in one organ, you start seeing aging and age-related diseases in other tissues like the brain and the muscles, for instance. And indeed, this caused the mice to become frail. So just by affecting one organ, having the bad neighbors in one organ, the entire mouse aged. This is, of course, phenomenal to me. You know, this means we can really target aging by targeting all these senescent cells in multiple organs in the body. And indeed, senescent cells have been shown to be culprits in many age-related diseases. So there are too many to name, so I group them in 3 classes, which we study in my lab as well. So metabolic diseases, we primarily focus on the liver, but also on the kidney, for instance. You see things like cirrhosis that is very much enriched for senescent cells. Neurodegenerations, diseases like ALS or Alzheimer's, whereas Alzheimer's, of course, is a disease of protein toxicity, but we know that causes senescence. And if you remove the senescent cells, mice do better. And musculoskeletal disorders, so for instance, muscle wasting, loss of bone mineral density, and osteoarthritis. And again, there's too many to name. But in all of these cases, mouse models for those diseases have been shown to do better if you remove the senescent cells. So why are the senescent cells bad? Because they have a 24-hour secretion of factors that are deleterious. It's low level, but it's there all the time. See it as a drug addiction. One time something can be good, but if you have it throughout life, it's actually very bad. And these factors are mostly pro-inflammatory, and this leads to the notion that senescent cells are what we call contributors of aging, so aging and inflammation together, and that makes them the root of the problem. And of course, I showed you that on a short distance, but also a long distance, they cause problems for health. Okay, I'm not presenting you this as if it's the final state. I am in a hospital, not in a university. So in a hospital, we try to do things to help humanity. So there's hope, and that is why I'm here. So this experiment is one of the most brilliant experiments I think was done, not by myself, but colleagues in the US. And what you see here is the lifespan of normal mice in the lab. So C57 Black 6, that's a mouse strain that a lot of labs use. 100% of the mice are alive at 1 year of age. And at some point they start dying off until at 2.5 years of age, there are not too many mice left in there. In fact, they all die. What these people now did is they made a genetic kill switch, which you cannot use in humans, but it's a trick they can use and do in mice, to remove these senescent cells once a month. And if you remove the senescent cells once a month, what happens? You don't change the maximum lifespan of the population at all. It's almost the same. But the median lifespan, so at 50% survival, you see an enormous switch. And this is actually more than 25%. So you can remove senescent cells in a safe way. This is what they show. And second of all, it has a huge benefit to health. And not only do they see this on lifespan, but also if you look at how the mice die, the mice here that go, they're dying in a healthy state. This is to me utopia. That getting old, we can do, but getting old in a sick state, nobody wants. But it's doable by removing senescent cells that you get mice at least to die in a gracious way. So now the question is, can we do this with compounds? And this is what I took upon myself already 7 years ago. I still pride myself actually on being the first in the world to actually make an anti-senescence compound. There are others that repurpose drugs, but I was the first one to design, a senescence compound. And I did it by the following rationale. If you now have a cell that was irreparably damaged, it can become senescent, as we've seen, but these are cartoons. If you now look at a real staining of a senescent cell, you see here the DNA with all the damage. That's what you see over here. So that blue is DNA. You see those little black holes in it. Those are condensed DNA, so the heterochromatin. And you see these red and green blobs. Those are the guardians on the DNA. So the DNA is damaged. Those are those real dots over here. You see them here in real example of a staining we did. And what we identified that in these, so these are complicated names, but they are basically markers for DNA damage and for activation of the two guardians, FOXO4 and P53. They are the guardians that prevent those damaged cells from dying. And I identified, and this actually is a movie, it should be rotating, but unfortunately it doesn't work here. But you have to take my word for it that we identified how the green FOXO4 is connected to the other guardians. And by understanding how that works, we could make compounds ourselves to destroy this complex. And we were successful in doing so. And that is indeed in 2017, we were able to show that we could kill these senescent cells, and doing this in aged mice. This is the same animal before and after administration of this anti-senescence peptide that I made against the two guardians. You remove the senescent cells, and you see that the mice did better. This was also widely presented in mainstream media, and of course it was very nice to get a lot of attention, but we also hit a dead end, because how do you get this to humans? This is what I want. I want to get this to society. And then of course you have a problem, because the European Medicine Agency, or the Food and Drug Administration in the US, they won't allow you to do these kinds of studies in humans. So we have to go actually for a real disease. So in short, can we develop therapies against senescent cells? Proof of concept is yes, and not just us. By now, there are a lot of groups that have come up with anti-senescence compounds. We show that at least breaking FOXO-P53 senescence has an improvement, has a beneficial effect on health of aging mice. And we are now trying to make this even better through commercial entities, for instance, to get it to society. And then, like I said, we hit this dead end because there's no way I can get this into the clinic in healthy volunteers that are 70 years of age. So we decided to go for a disease. And then I have to make a small jump to cancer, not because I don't like it. In fact, I love it that there's so much similarities between senescence and cancer. As I told you, what makes a healthy cell senescent? Irreparable damage. Cancer cells usually become cancer because they have so much damage. That is the reason that they become cancer. And in fact, so actually a lot of late-stage terminal forms of cancer show senescence markers. On top of that, how do we treat cancer? Aside from immunotherapy, it's usually with chemotherapy and radiotherapy, which I told you gives more damage. So if you already have cells that are expressing senescence markers, the chemo and the radiotherapy gives them even more of it. So that if they don't die from the chemotherapy, they become hypersenescence, if you will. So this is an ideal study system for us to test our compounds. In a nutshell, we tested several types of cancer, and I'm just showing you two here. So one of them is colon cancer, which in Utrecht we do a lot, so that's also one of the reasons why we focused on this. But this is terminal colon cancer, stage 4. We implanted that in the mice, in the colon, and you can see that the whole mouse at some point starts to express the tumor. So the signal here comes from the light that we gave the cancer cells. So it's called bioluminescence, with a difficult word, but it means the cancer cells, they expose light, and which you can measure. So this mouse is filled with the human cancer cells. And now if we apply the next generation compound that we designed, we can nicely reduce the tumor burden by a lot. And what I'm very excited by is now, if you look at different models, a triple-negative breast cancer, there are many compounds now effective against breast cancer, but not for this very difficult-to-treat group, triple-negative. No hormonal therapy really works over there. And almost all patients that are triple-negative die from the disease. And why is that? Not because of the primary tumor, but because of the spread of the cancer to distant organs, so liver and lung. These are called metastases. Now, if you implant a triple-negative breast cancer in the breast of the mouse, so the fat pad, indeed, you get metastases to the liver and lung. And look what happens with our next generation FOXO4 peptide. We can all but completely wipe out the metastases. Not so much the primary tumor, but that's not what the patients die of. The surgeon can remove the primary tumor, but it's really these metastases that are difficult to cure, and we can do that. So this is something which we are now trying to, through our startup company, to get into the clinic here in the UMC Utrecht. So can we target senescence in aging and cancer? I want to go for aging and age-related diseases, but it's difficult to do. However, for cancer, yes, because senescence markers are often elevated, especially in late-stage cancer, which is what people die of, not first stage, and those we can effectively target with our compounds. Okay, what's next for us? And now this is actually the fun part for me, because now I'm going to ask you, forget about everything I just said, because I think there's no such thing as senescence. It does not exist. So it's like saying cancer. There is no such thing as cancer. You have a stage 4 CMS2 type colon cancer. So you have all these better definitions. But for senescence, the field is extremely young and naive in that regard. We still say senescence and SASP as if it's one thing. So this is the current view. So this is again the cell that I've shown you now many times. The cell that is senescence, it secretes a whole range of things, the SASP factors, and that does all these age-related diseases. In fact, there are now, like I said, about 50 compounds that people present as anti-senescence compounds, and they're all claimed as golden bullets. So you kill the senescent cells, they remove, and they undo all the aging-associated diseases. It's not true. I refuse to believe that. Instead, this is my model, and I think it's very important that, for instance, this cell, the senescent cell type 1 that secretes the red and yellow, it will lead to a different disease, different pathology than the cell that has the green and the blue factors secreted. It sounds obvious, right? But it is in the field not at all acknowledged. And of course, then you can say, well, that's a nice theory, but is there any evidence? So why am I doing this? Because I don't believe of these 50 compounds that are now developed that any of them will make it as a golden bullet in humans. I refuse to believe that. We need to much better define when to apply which compound for which specific age-related disease. And so we can make more precise therapies. And then of course you can say, well, that sounds nice, it's a theory. Okay, do you have any evidence for this? Indeed, I do. So here we have a culture of senescent cells. So this is directly from the lab. It's again a staining we did. So in blue, you see the DNA of these senescent cells. So each of these is a cell. And I stain them with two very famous markers for senescence, the green and the red. It doesn't matter what they are, although they're named over there. You can see that they're never in the same cell. So if I would do a population analysis of this dish, I would say, yes, there is senescence because the green and the red marker are there. But if you look at single cell level, this is not the case. That means that I'm at least partially right. There are subtypes of senescence. And there's more evidence to it. So now we actually have real-time evidence that, for instance, you see 4 different examples of senescence markers that in this experiment, this green cell can turn into a red cell the day after. So these are things that are dynamic. The location also matters. So again, these are senescence markers, but depending on where you are in your experiment, you have the marker or you don't have this, and so on and so forth. So we need to much better understand when we have which particular population of senescent cells in the body, and then, because that means that we can develop compounds to target them. So then the last part is, can we use this knowledge to make better therapies for senescent cells for cancer? For humans, and this is what I hope to do here at the UMC Utrecht. And to do that, we've done it already for one of our types of senescent cells, so the FOXO4 peptides. We apply advanced 3D structural imaging to make 3D structures, and this has led us to the following: that in 2018 to 20, we made 400 new versions of this compound. So we synthesize them, we produce them, and make sure that they have a better targeting for the deleterious types of senescence. And we are in the middle of doing these mouse experiments and trying to outsource the pharmacology and get them to the clinic. And then in 2024, the ambition is to actually start human trials here in Utrecht.